Welcome to my channel Adhyan with Flow, Chapter 13, Photosynthesis in Higher Plant. This is the name of this chapter. Now, what is photosynthesis? What do you understand by this term? Photo means light, and synthesis means something formed, something produced, something formed with the presence of light. Clear, guys? Clear this term? We will study photosynthesis in plants. Look at here. This is the stem. This is the root. This is the leaf, and this is the flower part. In plants, leaf perform what? Photosynthesis. Leaf perform what? Leaf perform photosynthesis. They form what? They convert light energy into chemical energy. What they do? They convert light energy into chemical energy. What is the structure of leaf? What kind of things they contain so they can easily do photosynthesis? I am writing here. This is the leaf dissect part. This is the cuticle layer. Stomata present for gaseous exchange. Here, epidermis, hypodermis. They contain some mesophyll cells. In mesophyll cells, they contain chloroplast, the green color organelle chloroplast. Chloroplast is a double membranous organ. Chloroplast is a double membrane structure. In chloroplast, they contain what? Thylakoid. These are the coin-like structure. And thylakoid contain what? Chlorophyll. Yes, guys. So this pigment, what they do? They do photosynthesis in plants. Look at here. This is present in their higher security. Light penetrate here, then here, then here, then here. What kind of chemical energy is formed? We know now light energy converts into chemical energy. And what kind of chemical will form? Yes, we call it as a food. C6H12O6. The synthesis form of food is glucose. Now, the transport form of food, we call it as sucrose. The storage form of food, we call it as a starch. So this is the synthesis form. Plant make glucose, and if plant supply the whole body part their food, this form circulate in the plant body. If they want to store the food, this form they store. Clear, guys? So this statement is all green plants, what they perform? They perform photosynthesis. In fact, some cyanobacteria also perform photosynthesis because they have chlorophyll. Now let's move further. There are some experiment, guys. Now let's begin with experiment first. I'm talking about this experiment. In this experiment, what kind of experiment scientists will do? What they do? They take KOH. Why they take KOH? Because it absorb what? CO2. Okay, this one thing is clear. In this experiment, scientists take CO2 absorbing KOH. KOH, potassium hydroxide, useful for absorbing the CO2. Now scientists, what they do next? They take a plant leaf. And half portion is directly contact with light. Is this term is clear? Now this half portion covered by some apparatus, and here this KOH is present. Now what happened next? The scientists watch that This part of leaf will remain green, will remain green. But this part of leaf, they are not green. They are not green. It means scientists will understand with the presence of light, with the presence of light plus CO2, water. These are useful for what? These are useful for making food. Yes. So light is, yes, here. Now water is from the base part of the roots. Okay. Now CO2. What happened with CO2? CO2 absorbed by KOH. So plant can't take CO2 because all CO2 absorbed by KOH. So plant can't make any kind of food, any kind of starch, any kind of glucose, nothing. So this is the first experiment that CO2 somehow important in food production, because all kind of CO2 absorbed by KOH. So this part of leaf can't get any CO2. Let's read here. Let's read here. A part of a leaf is enclosed in a test tube containing some KOH soaked cotton. Here this is the enclosed part of the leaf, and here is cotton soaked by KOH is present. Here is KOH is present. Now the other part of half exposed to air. Look at here, this part is exposed to air. Clear? The setup is then placed in light for some time. Yes, the setup is present in light. On testing for the presence of starch later in the two parts of the leaf, this part make starch. Why? This part makes starch. Why? Because from surrounding CO2 consumed by this portion of plant leaf, light is also present and produced water is taken by the roots part. Okay, water take by root part. But here, what happened with here? Here this leaf part also exposed in light. They also get water, but they don't get CO2. Why? Because whole CO2 absorbed by KOH. So this is the first experiment that CO2 was required for photosynthesis. So this one thing is clear, that CO2 is required for forming the food. Now next experiment, Joseph Priestley's experiment. What they take? They take jar. They take bell jars. What they do? What they do? First, step A. They take mouse and a candle. Candle release some of the foul air, bad air that is consumed by rat. So rat die. Now another time, what they do? They take again rat. They take again a candle, and they take a mint plant. They take a mint plant. Now mouse is still alive. What they distinguish? What they distinguish? What they found, that candle release some foul air in atmosphere, and this foul air is absorbed by plant leaf, and this plant leaf release some good air, and this is taken by this mouse. So mouse can still alive. So this is the experiment of Priestley. Priestley observed that the candle burning in enclosed space, a bell jar soon get extinguished. Similarly, a mouse would soon suffocated. Of course, if they get bad air, then they feel suffocation. In closed space. He concluded that a burning candle or an animal that breathed the air, yes, they breathe this air, but somehow damage the air. But when he placed a mint plant in the same bell jar, look at here, this is the mint plant, he found that the mouse stay alive and the candle continued to burn. Priestley hypothesis. Plant restored to the air whatever breathing animals, means plant release such kind of gas, those are beneficial for animal. They contain such kind of air that are beneficial for the animal, and still burning candle remove. Matlab, guys, this candle release some foul air. This foul air absorbed by this plant. Plant release some good air. This absorbed by this animal. That's it. That's all about this experiment. Now let's switch towards the next. Okay, next. Let me check. Some experiment also support them. Okay, some experiment also support this Priestley's experiment. Now another experiment is Jan. What they do? They take a water plant. What they do? They take a water plant. This is the water plant, and this is the another aquatic plant. Okay guys, this is the another aquatic plant. This is the water plant. This is the aquatic plant guys. They present in aquatic medium. What scientists do, what they do, this plant they put into light, and this part they put into the dark side. Later what they found, they saw that plant produced some air bubbles. Light light zone plant, they show some bubbles, small bubbles formed around the green parts of the plant. Yes, when scientists isolated this air bubble, they found oxygen. Means green plant release what? Green plant release oxygen. And this plant that scientists kept in dark, they does not release oxygen. They does not release oxygen. So scientists will conclude that plant take CO2 and they release oxygen. This experiment done by Jan Egenhauser. Now, in our first experiment, we get that CO2 is necessary for making food. So we identify that food is formed by CO2, means food. That plant produce food. In another experiment, we found that CO2 is harmful for animal, but it is beneficial for plant. In next experiment, plant release what? Oxygen, and they take CO2. This is the third experiment. And further, again, another experiment done by Julius and Sachs. Now what they do? They said that green plant have some green substance, some green substances. They call it as a chlorophyll, and they produce glucose. So this sequence given by Julius and Sachs, they said that plant contain green substance. Then plant contain green substances. Now we call it as a chlorophyll. Now this green substance, right now we call it as a chlorophyll. This located in special bodies. They said that this chlorophyll located in the special body. We can call it as a chloroplast. Here thylakoids membranes contain chlorophylls. They contain chlorophyll. So plant will produce glucose and plant store food as a form of starch. Now next experiment is done by T.W. Engelsman. Now what they do? They use prism. They use split light. And what they do? They take cladophora. This is the green algal plant. And now what they do? They thought this is the aerobic bacteria. This is the what? This is the aerobic bacteria. Now what happened with this? They want to measure that how much wavelength they need, how much wavelength light they require for what? For what? For photosynthesis. In what kind of wavelength they produce food? Means sun have multiple wavelengths light, 4 nm to 700 nm. What they do? Now what they do here? They take plant. The plant name is cladophora. The plant name is cladophora. Okay, they arrange this. They arrange this thing. Now, what they found next? They put some aerobacteria here. They put some aerobic bacteria. Okay, they put into their experiment. Now, what they found? This is the aerobic bacteria. Of course, they absorb oxygen. Now, what they found? That this is this cladophora, green plant. Okay, this is the green plant. This here is 400 nm light is exposed. Here is 700 nm light exposed. Now bacteria deposit here maximally, and aerobic bacteria deposit here maximum. So according to bacteria, we can easily identify that at a 400 nm and at a 700 nm. Here is also some bacteria present, but here and here they found in maximum amount. Here is prism light source. So scientists, what they conclude that at a 400 nm and at a 700 nm, the plants show highest photosynthesis. The plants show highest photosynthesis because that time plant release highest oxygen. Highest oxygen means at this time, at this wavelength, plant will do photosynthesis in their highest amount. in their highest amount. Then we isolate this cladophora, what kind of things they have. We found that we contain chlorophyll A and chlorophyll B. This is the very important chlorophyll. This is the center chlorophyll. So this is experiment of Engelsman. Whatever I explain here, already they are written here. Now next scientist will come. And this is now Cornelius and Neil. They are microbiologists, not a botanist, not a zoologist. What they demonstrate? They said H2O gives oxygen. Yes, when we study this reaction, in this reaction, yes, H2O, the water molecule gives future oxygen, not CO2 gives oxygen. Now H2O gives oxygen. And they prove, how they take isotope of water. They take isotope of water. Okay, and they analyze that here's oxygen also contain this kind of isotope. So this experiment will prove that oxygen gas come from H2O, means oxygen come from water molecule, because they tag oxygen with their isotope, and this isotope present in the oxygen. So they confirm that H2O will give oxygen, and CO2 they give food. Clear, guys? This CO2 does not give any kind of oxygen molecule. So H2O provide oxygen. So this experiment done by Cornelius von Neil. Cornelius von Neil. This is a single experiment. This is a single scientist. How they identify? Because they experiment in their lab and they demonstrated some bacteria, purple. What they absorb? They absorb some sulfur. They absorb some sulfur and they release sulfur gas. Okay, they does not absorb H2O. They absorb sulfur form and they release in atmosphere the sulfur. So H2O, this thing release oxygen in atmosphere. So this is all about our scientist. That's all about our scientist. And that's all for now. In our next chapter, in our next video, we'll start from here. And if you still not subscribe my channel Adhyan with Flow, then please subscribe, guys. Please subscribe. And thank you. Again, thank you for listening me. Keep smiling and keep revising. Thank you.